Welcome to Chapter 15, which is Evolution and Diversity Among the Microbes. So we are actually going to be going out of order from this point on as far as chapter number, because I like to start with the smaller organisms and then work up to the larger organisms. And this is actually really appropriate for why we're all doing our studies remotely at this point. So this section, we're going to be looking again at bacteria. So a little bit of review on bacteria, but some new information on that group. A group called the Archaeans. And then we're also going to be looking at the protists and viruses. So all of these have something in common, and that is we cannot see individuals with the naked eye. So before we can get into that, just a little bit of terminology, because you'll see these terms showing up, and so I just want to make sure that we are all on the same page as you hear these come up, both in my videos and a couple of videos I'm going to be linking in this series of lectures. So when we organize organisms, we like to be able to do this so that we can show their relationships, and so you're looking at basically the giant family tree for all life. And this is organized showing the domains of life. Domains are the biggest groups that we can put living organisms into. And so what you're seeing here is bacteria, archaea, so the two groups that I just mentioned that we'll be talking about in this chapter. These are each their own domain, so they have their own branch on the living tree. And then the other branch set, which is a whole bunch you can see over here: protists, plants, fungi, animals. There's a bunch of little branches, but they're all in the last domain, and that last domain is the eukarya. Okay, so the one that we're going to be talking about here with the microbes are the protists, which you can see are sprinkled all throughout the tree, and we'll talk a little bit about why that is as we get farther into this chapter. So the thing is that there are microbes in all three domains. So you can see bacteria and archaea have their own branch, and then protists are scattered throughout the eukarya. And you might notice eukarya sounds a lot like eukaryotes, and that's because all of the organisms that are in the domain of eukarya are made up of eukaryotic cells. So here are the ones that we're going to be looking at. So domain bacteria, domain archaea, and then domain eukarya. We're going to be focusing just on the microbes in this chapter. So this is being an example of that. A giant amoeba. This is one of the single cells that you may be able to see with the naked eye, but it would just look like a speck of dust in water. Now you'll see that the fourth group that we're going to talk about, viruses, they're not on, sorry about that, they're not on the tree of life. They aren't currently considered to be living organisms because they don't have any of the characteristics of life, which we'll talk about later when we get into talking about these guys. And so they're not put on the tree with everybody else. But they are here. We will be talking about them because they do affect living organisms. So the thing is that these microbes are going to be the simplest organisms on the planet, but they are the most successful. They are the most numerous organisms on the planet, and by weight, we are outweighed by our microbes. So if you were to calculate how much there is in like bacteria and archaeans and protists, we are well outweighed by those microbes compared to the eukaryotes. And as I mentioned, they're tiny, super, super small. So here to give you an idea of their relative size, I mentioned that the amoeba is the one that you may be able to notice a single cell with your naked eye. Like I said, it would look like a speck of dust. So this is one of the biggest guys at 1 millimeter across. So this guy, if we were to make these guys relative to objects in the visible macro world, this amoeba would be the size of an oak tree, And then the guys down here at the other end, an influenza virus, would be the size of a pinhead. Basically like your writing pen or pencil. The very tip of that would even be smaller than that. So you can see the viruses are incredibly tiny. So this is nanometers. This right here stands for micrometers, that little weird kind of M shape. And so E. coli, a little bit bigger than flu, the influenza virus. And then bacteria, we saw these guys when we were doing microscope work. And we had to view them on the highest power that our microscopes had. And then Euglena, which is one of our protists. A little bit bigger, we would have had to view this at the 400 power on the microscope. So not the highest power, but almost the highest power to be able to get some good detail. So we have a wide variety of sizes. We also have a lot of different species in the microbes. Right now we have about 500,000 different species identified in the microbe groups. So that makes them very diverse. However, we expect to actually identify millions more. The problem is that a lot of these microbes look very similar to each other. And so we can't actually figure out what some of them are until we do things like genetic testing. And so, especially with things like bacteria and archaeans, we're only now finding new species as we're able to run genetic tests on them to distinguish species from each other. The other problem is that we'll see some of these species show up in the weirdest places. And so as we are discovering more of those areas, we're finding more of these organisms, because they can live almost anywhere and eat almost anything. Members of the bacteria and the archaeans especially have been found everywhere. So we have members from these groups that live, for example, inside of other organisms. So E. coli is one of the ones that populates our intestinal tract. And we're not the only organism that is populated by other bacteria or by bacteria. And they will eat just about anything. We'll see there are different types of food for bacteria where they can eat things like chemicals, and same is true for archaeans. You can find them in almost any environment. So here, this one, Thermus aquatus, this one is a archaean that's actually found in the hot springs at Yellowstone National Park, and that water is above boiling. So that should kill just about anything. These guys are there. What you're seeing over here, it looks like waves, but this is actually salt at the Great Salt Lake that's evaporated out. The water is over here. These are halobacteria, or salt bacteria, that are able to live in this very salty environment. So find these all over the place. There are bacteria that live in the Arctic and Antarctic. So they live at very low temperatures, obviously very high temperatures with the hot springs, low pH, high pH. I mean, we find these just about anywhere you can think of. Bottom of the ocean, hydrothermal vents, underwater volcanoes. So we've got these all over the place. So the first group that we're going to focus on is the bacteria. Actually, out of all of the microbes, these might be the most diverse of all of them. They're very numerous. We find them everywhere. So what are bacteria? This is a little bit of review for this section. So what I want you to do, I will also be posting up a video that has some of this information in it. It actually kind of walks you back through the structure of the bacteria. It's got also some examples of the different shapes. These are the three most common shapes of bacteria. So you've got spherical, or cocci, or coccus for round. We've got bacilli, or bacillus. These I think of as tic-tac shapes. These are the rod-shaped bacteria. And then spirilla, or spirillus, for spiral shapes. This is one way we can actually classify bacteria. The other thing that will be in that video, and you should also have it in your notes, you want to review how bacteria reproduce. How else can we identify bacteria? Like I said, we can do genetics, but there are some quick and dirty ways to identify bacteria by species. So we can do it by their shape, but the thing is that there's only a limited number of shapes. And they're all very similar in size. So another way we can do this is looking at the colors and shapes of their colonies. So if we sample bacteria and use these petri dishes, what's in these petri dishes is different types of food. It's basically kind of like Jell-O. It is not the kind of Jell-O that you would be normally eating, but the material is very similar to Jell-O. And we can put different types of food in it. So we can put things like glucose or fructose or sucrose. We can also put things like some bacteria feed better on things like blood. I know that sounds really gross, but we can put these different materials into this food material, this agar, this Jell-O type material, and then we can swab the bacteria across the surface and they will start to grow. Now we can't see the individuals, but what we can see are the colonies, and that's what you're seeing here. So you can see, for example, this group, they grow in these little tiny dots, kind of spaced all over the place. In small groups, while this one down here, they grow into these bigger dot groups, while other ones are just going to grow kind of in solid areas. Other ones will grow into lines, and you can see that there are different colors. Now some of these colors are because of the agar that's behind them, so this one's actually kind of pink behind it, but the actual bacteria group is pink. So this color of what they actually grow into can be used to identify them. There's also a process called gram staining. And what gram staining does is it actually is a process of a set of dyes that we use to dye the bacteria. And what it'll tell us is if in the cell wall there's a specific chemical called glycoprotein. Basically, we're looking for where that layer is. So this gram stain process will tell us where that molecule is in the bacteria. So gram positive says that it's outside of the cell wall. Gram negative says that it's actually underneath of another membrane. And so it's going to be gram negative and it's going to have this pinkish reddish color. So why do we care? Well, this actually makes a difference on what kinds of antibiotics will work on that bacteria. So you can see here, gram-negative bacteria, so the ones that turn this red-pink color, are actually going to be resistant to the group of penicillin type of antibiotics. So we're going to have to use something else on them. So if you've ever had a doctor take a sample thinking you have a bacterial infection, saying they're going to send it out to the lab to verify, that's what they're usually doing, is making sure, okay, we think you have strep, but we want to make sure if it's positive or negative. It's because they want to make sure they're giving you the right kind of antibiotics to treat it. So at this point, I'm going to stop this lecture. Like I said, I'm going to put up a video from the Amoeba sisters, which is going to kind of review some information. It's going to have information on reproduction, parts of the bacterial cells, and so forth. And then I'll pick back up with more information on bacteria after that.